Hi, this tutorial is the third in the Cellular Respiration series. In this tutorial, we'll be discussing the TCA cycle. The TCA cycle is also known as the citric acid cycle or the Krebs cycle. So let's jump right in. The primary substrate for the TCA cycle is pyruvate. Fatty acids and amino acids can also be used, but we're not going to discuss them in this tutorial. Pyruvate is produced by glycolysis, and the first thing that happens to pyruvate is that it gets converted into acetyl coenzyme A, more commonly known as acetyl-CoA. This conversion happens via the action of three enzymes, collectively known as the pyruvate dehydrogenase complex. When you see the word complex after an enzyme, it usually means there are multiple enzymes involved, but they are functionally considered as one. As we saw in the glycolysis tutorial, dehydrogenases are the enzymes which produce molecules like NADH and FADH2. Accordingly, the pyruvate dehydrogenase complex produces an NADH molecule. Now I've drawn the acetyl-CoA as two balls of carbon. This is because acetyl-CoA is a two-carbon molecule. In this tutorial, we're going to pay special attention to the carbon balance throughout the TCA cycle. Hence, it is easier to illustrate the molecules involved in this way. Note that acetyl-CoA has one less carbon than pyruvate, which is a three-carbon molecule. The extra carbon is lost as carbon dioxide as a byproduct of the conversion to acetyl-CoA. So we'll look at the molecules involved first, and then we'll discuss the enzymes involved. The TCA cycle begins when a four-carbon molecule known as oxaloacetate combines with acetyl-CoA to produce a six-carbon molecule known as citrate. Citrate becomes the six-carbon molecule cis-aconitate, and then cis-aconitate becomes the six-carbon molecule isocitrate. In the next step, we lose one of these carbons as carbon dioxide, and we also produce another NADH. Thus we are left with a five-carbon molecule known as alpha-ketoglutarate. The same thing happens again. We lose a carbon as carbon dioxide and gain an NADH. Hence we are left with only four carbons in our main molecule, which is now called succinyl-CoA. Succinyl-CoA is then converted to succinate, and a GTP is produced in the process. GTP is essentially an equivalent of ATP. Succinate is then converted to fumarate, and an FADH2 is produced in the process. Fumarate becomes malate, and malate completes the cycle and becomes oxaloacetate, producing the final NADH in the process. The oxaloacetate is then available to combine with any available acetyl-CoA and begin the whole process again. Now let's take a look at the enzymes involved. Citrate synthase predictably synthesizes citrate from oxaloacetate and acetyl-CoA. Then, the enzyme aconitase produces cis-aconitate and then the conversion back to isocitrate, which is a structural isomer of citrate. Again we see dehydrogenases in action, with isocitrate dehydrogenase producing the NADH in the isocitrate to alpha-ketoglutarate conversion. And the alpha-ketoglutarate dehydrogenase complex also producing NADH in the conversion to succinyl-CoA. Succinyl-CoA synthetase catalyzes the conversion of succinyl-CoA to succinate. Then yet another dehydrogenase, known as succinate dehydrogenase, produces the high-energy molecule FADH2. Fumarase converts fumarate to malate. And then the final enzyme is another dehydrogenase, malate dehydrogenase, producing NADH and converting malate to oxaloacetate. And that's an overview of the TCA cycle. If you've enjoyed this tutorial, please help us produce more by making a donation at www.handwrittentutorials.com.